In this video we will discuss about hemochromatosis pathogenesis. So this hemochromatosis disease is associated with the iron. It is mostly because of overloading of the iron within the body that might cause several diseases or several problems. So let us first understand how this iron content is maintained within the body. So uh, one of the protein that helps in maintaining this iron content is hepcidin. What hepcidin does is hepcidin binds to the FPN, or we can say ferroprotein, and this is the receptor, or this is the channel by which iron comes iron comes from within the cells into the extracellular matrix. So this hepcidin binds to the FPN and uh, transport of cells from within the prevent the transport of cells from the cytosols of the cells into the extracellular matrix. What happens when hepcidin binds? It will then divert it to the lysosomes, through early lysosomes, then late lysosomes, then lysosomes, endosomes, through early endosomes, then late endosomes, then lysosomes. However, hepcidin is not only the factor that controls the overloading of the iron. There are other factors also, such as BMP6, HFE, HJV. So how they interact is that whenever there is a high content of this iron, either free iron or this TF bound iron, this BMP6 will bind to the receptor, and it will triggers the activation of hepcidin, and BMP6 and HAV will compete each other in a way that this HAV will compete each other in a way that whenever this soluble HAV binds to the membrane it will trigger the activation of hepcidin in different we can say in different degree compared to the BMP6, and simultaneously this HFE will also, when bind to the TFR2 and TFR1, it will triggers the signaling pathway that will trigger SMAD158, and this will finally leads to activation of hepcidin. And one more thing that has been identified so far is that higher iron content within the extracellular matrix will somehow trigger SMAD7 that will inhibit that will not higher we can say higher iron content within the cells, not in the extracellular matrix. Higher iron content within the cells will somehow triggers SMAD7 that will inhibit this hepcidin formation, so that iron can go outside. However, how this thing will happen is not yet completely known. So let us now understand uh, in big picture how this hemochromatosis happens. We will start with the normal functioning. Normal functioning, this hepcidin is released from the liver in normal case. If this HJV, HFE are functioning, HFE, and FPN are functioning normally. FPN is ferroprotein. So this hepcidin will binds to the ferroprotein receptors and prevent the overloading of iron content in the extracellular matrix. In the panel B, of the right figure shows the mutant HFE condition. In this, what happens is that hepcidin formation reduced, and because of that, more iron is released from macrophage and these enterocytes, and this will leads to uncontrolled release of iron because of less amount of hepcidin formation, and with time iron will accumulate in the extracellular matrix and get overloaded. And in case of this HJV is not functioning properly, or HJV is mutant, the hepcidin is reduced to a very severe extent, such that the overloading of iron within the extracellular matrix is reaches to a very high level, and even higher than compared to HFE mutant condition. The last condition is that if even though all the proteins that are responsible for hepcidin formation are functioning normally, it might still cause the hemochromatosis problem. And this condition might occur when there is a mutation in the ferroprotein or FPN. So what happens in this case is that ferroprotein become unresponsive to the hepcidin, and it will continue to export iron in the extracellular matrix even though it has a sufficient amount of hepcidin. So this condition is very rare compared to the other two conditions. However, this condition is still also exist uh, in some patients. So I hope you have understood something about how something, or we can say, pathogenesis of hemochromatosis, and what are the different kind of hemochromatosis diseases exist nowadays or in the world. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.